Okay, in science, there are different microscopes that are used. They have different functions. Scientists would use different types of microscopes depending on what they needed to look at. The compound light microscope, or sometimes just light microscope, this is the microscope that we would use in our classroom. The difference, this one is showing you two eyepieces. The microscope in our classroom, we only have one eyepiece. They're used to view wet mounts of living organisms or prepared slides. A wet mount means you have taken a sample of something such as a tissue and you add water and sometimes a stain to that substance so that you're able to look at it. The light microscope produces a two-dimensional image and it can magnify up to 1,000 times the actual size. Now the magnification is going to vary based on the different objective lenses and then the magnification within the eyepiece. Our microscopes in the classroom do not go to 1,000, however they do have the capability if we were to change those lenses. This is an Eladia leaf that is looked at under a light microscope, just to give you an idea of what it looks like. An Eladia leaf, many of you are probably familiar with the green, some people would call it seaweed, but it's not, but the tall green plant that you would find in a fish tank, that is Eladia. The scanning electron microscope, known as a SEM, it's going to send a beam of electrons over the surface of the specimen that has been dehydrated and then coated in gold or platinum and sealed within a vacuum. This is going to produce an image that is three-dimensional. This way they're able to look at the structure and surface of the specimen. A scanning electron microscope, or SEM, is able to magnify up to 100,000 times. This is an example of an ivy leaf underneath a SEM or scanning electron microscope. You can see that you're able to see things in much more detail here. There's a transmission electron microscope, the TES or TESS. This sends a beam of electrons through a thin slice of a sample, and then it sends the image to a fluorescent screen. It's going to produce a two-dimensional image. It's a projection of the sample. It can magnify up to 2 million times. This is taking a look at an animal cell underneath an electron microscope. Again, you can see how it's able to see many more structures. This is just a comparison. This is showing you again that Eladia leaf. The Eladia leaf, looked at it underneath the light microscope versus looking at it underneath an electron microscope. Each type of microscope has advantages and disadvantages for viewing specimens, and each creates a unique view. The images below show plants as seen under 3 different microscopes. The SEM shows the surface of the leaf with a close-up of a trichome. This is the trichome. The TEM shows the chloroplast of a leaf, and then the light microscope can show the individual cells within a leaf. The biologist has to choose the microscope based on what observations they need to make. So again, looking at the leaf, you have your chloroplast shown underneath the transmission electron microscope, and then the individual cells are seen on a light microscope. The last microscope we'll look at is the dissecting microscope. Dissecting microscopes, these are designed to be used at a low magnification. They're typically using light reflected from a surface or an object rather than transmitting light through something. Larger objects can be viewed in a three-dimensional. The samples don't have to be sliced. They can be larger samples, and they can actually even be a live sample. The light can be passed through from underneath the sample, but it is also comes from the top, or if you have an external light source, they can use that on the sides. A dissecting microscope is able to magnify up to 100 times. Here's an example where the dissecting microscope has been used, where they're able to actually look at the little organisms. With the development of the microscopes, this allowed scientists to look deeper into the cells of organisms, leading to the cell theory. Discovering the cells. In 1665, and you guys don't need to know dates, it just kind of puts it in perspective of how things played out. Robert Hooke discovered cells while he was looking at a thin slice of cork tissue. He described the cells as tiny little boxes or honeycombs, and that's where he came up with the term the cell. However, he believed that the cells only existed in plants and fungi. In 1673, Anton von Leeuwenhoek, he used a handmade microscope to observe pond scum. He discovered single-celled organisms within the pond scum, and he gave them the name animalcules. After that, there was a 150 to 200 year gap. There was a period of time where the scientists believed in spontaneous generation. For example, mice grew from dirty clothes and corn husk, or maggots were able to just grow from rotting meat. Louis Pasteur is the scientist who receives credit for disproving spontaneous generation. There were some other experiments before Pasteur's, but they had some flaws. Pasteur's experiment, he used flasks containing the broth solution, but his flask had these swan-necked or curved-necked openings. This allowed for air to enter into the broth, but any dust or microbes would get stuck in the curved part of the tubing. You see where the dust particles get stuck here. He heated the broth, but nothing happened. There weren't any microbes appearing within the broth solution. Even after weeks, nothing was appearing within the broth solution. So then he took some of the flask and he actually broke off this curved part of the tubing, and then within a few days the broth solution became cloudy. This indicated that those microbes that were appearing had to come from something in the air and not just spontaneously appear within the broth. Continuing to look at cells, we can develop the cell theory. We looked at Robert Hooke and Anton von Leeuwenhoek. Now we have Schleiden in 1838. He concluded that all plants have cells. And then in 1839, another scientist by the name of Schwann discovered that all animals have cells. Therefore, all living things would, because the other scientist, Schleiden, had already found out that plants did. This is kind of a silly analogy, but to kind of keep this straight, who did plants and who did animals, I think Schwann, like a swan, and a swan is an animal. And then in 1858, Virchow concluded all cells come from other cells. And this leads to the cell theory. There are 3 parts to the cell theory that you must know. All organisms are composed of 1 or more cells. The cell is the basic unit of structure and organization in organisms. All cells come from pre-existing cells. And then just some couple interesting facts about cells. The average human being is composed of around 100 trillion individual cells, and it would take as many as 50 cells to cover the area of a dot on the letter I, just to kind of give you an idea of how tiny they are.